Okay, chapter 17, Mechanics of Breathing. So, in this chapter, we're going to talk about the respiratory system. We'll discuss gas laws and then ventilation, which is basically the movement of air. So, the respiratory system has four major functions. The first being it's where you're going to exchange your gases. So, the exchange is going to happen between the atmosphere and our blood, obviously through the alveoli. Now, we're going to try to bring in oxygen and we will breathe out carbon dioxide. Another function of the respiratory system is that it regulates your body pH. So, the major organ we talked about that really responds to changes in pH after buffers was our respiratory system. And the respiratory system will do that by selectively either retaining or excreting carbon dioxide. So, again, carbon dioxide in your body is essentially like having acids in your body. The respiratory system also is important for protection. So, it's going to protect you from inhaled pathogens and irritating substances. So, the mucus within our respiratory tract will try to trap those. And this, with the help of cilia and with the help of coughing, we'll try to get it out before it actually gets deeper down into our alveoli. And it's important for vocalization. We wouldn't be able to speak or make noise without the movement of air. Now, let's take a look at the principles of bulk flow of air. Now, some of these concepts will sound familiar. For example, air will only flow from areas of high pressure to low pressure. So we have to have a pressure gradient. In our respiratory system, the muscles that make up our respiratory system help create that gradient, particularly the diaphragm. And air flows through tubes, so our airways. So resistance to airflow is going to be influenced by things like our tube diameter. So in lab, when you did cardiovascular dynamics, you learned about how vessel radius influences resistance and blood flow. Well, the same concept applies here for airflow. So the respiratory system, we can look at external and internal respiration. So external respiration is the movement of gas between the environment and our body cells. So air coming into the airways, into the alveoli, circulating in our circulatory system is all considered external respiration. Internal or cellular respiration is when we use oxygen. And what do we do with oxygen? We use it for aerobic metabolism to make ATP. And in the process of making ATP, we create carbon dioxide. And that's why we have to expel carbon dioxide because we are creating it. So our blood will pick up the CO2, eventually send it to our lungs where we can then expire CO2 in our air. Now, the structures of the respiratory system, we have our conducting system. Those would be the airways that help conduct or move the air into our lungs. So they would include things like our nasal cavity, our trachea, our bronchial tree. At the end of our bronchial tree, we have our alveoli. So the alveoli are the little sacs where actual gas exchange takes place. And the other structures of the respiratory system are made up of the bones and muscles of the thorax. And these are going to assist ventilation. So, for example, our diaphragm. Without our diaphragm, we really wouldn't be able to breathe. So, what are some of the muscles? So, we mentioned the diaphragm, but there are more. Okay, so let's talk about ventilation and the muscles by separating the muscles that are important for inspiration versus expiration. Now, before we get into that, let's make sure we understand quiet breathing. So, quiet breathing is essentially normal breathing or passive breathing. So, this is the breathing that you do normally when you're just sitting there listening to this lecture. So, this is quite different than active breathing. So, active breathing is when you take those deep breaths. So, during quiet, normal breathing, during inspiration, we will contract our diaphragm and our external intercostals. Now, when we have to do deep inspiration, what we'll do is we'll still contract our diaphragm and the external intercostals. We'll just contract them more because these are skeletal muscles. We can contract with greater force. In addition, we will recruit our scalenes and our sternocleidomastoids to help assist our inspiration. So it helps make our rib cage, it expands it and helps expand our lungs essentially. Now, during quiet expiration, we actually don't contract any muscles. What happens is, during quiet inspiration, we contracted the diaphragm and the external intercostals. During quiet expiration, all we do is we relax those muscles. So the recoil of these muscles relaxing is what helps collapse our lungs and helps us expire air. However, if we have to do deep expiration, let's say we're trying to blow up a balloon, then we would recruit and utilize our abdominal muscles and our internal intercostals. Now, our lungs are surrounded by a sac. We call that our pleural sac. So here is our lung, here is our pleural sac around it. The pleural sac has two membranes. We have the inner and the outer membrane. The inner membrane is attached to the lungs. The outer membrane is attached to the thoracic wall. Now, between the two membranes, it's filled with pleural fluid. So what is the purpose of this pleural fluid? Well, number one, it creates a moist, slippery surface so that the opposing membranes can slide across one another as our lungs move. So if you think of this blue air sac as a balloon, imagine if you were to blow up this balloon, this pleural sac that's around it will have to expand. And when it expands, the two membranes will start to slide against one another. If we didn't have pleural fluid, the friction that's created would actually cause the pleural sac to burst. And so that pleural fluid creates the slippery surface so that the membranes can slide past each other without the fear of it bursting. The other thing that pleural fluid does is it holds the lungs tight against the thoracic wall. Essentially, the pleural fluid is acting like glue. So this inner membrane that's attached to the lungs and this outer membrane that's attached to the thoracic wall are held together by the pleural fluid. Because what happens is our lungs actually want to collapse. Our rib cage actually wants to expand. The reason our lungs don't collapse and the reason our rib cage doesn't expand is because the membranes are holding them against one another. And so when we have, let's say, a pneumothorax and this pleural sac is compromised, that's when our lungs will actually collapse because the glue is no longer working. Okay, so let's take a look at our bronchial tree. So our conducting system, again, is made up of our airway. So we have our trachea, we have our bronchial tree, and notice we have these blue C-shaped cartilage rings. And what these do is they keep your airways open. And by keeping your airways open, preventing them from collapsing, it makes it much easier to move air. So it makes it much easier to ventilate. So we don't have to work as hard to breathe. Now, our bronchial tree will branch many, many times. And every time it branches, we're able to increase the surface area of gas exchange. So we actually have like 23 branches. At the end, we have our bronchiole. Now, bronchioles are where we will bronchoconstrict and bronchodilate. Now, these bronchioles are not surrounded by the C-shaped cartilage, which is why it can constrict and dilate. And at the end, we have our alveoli. And our alveoli is where actual gas exchange takes place. Now here's a blow up of our alveoli, and what I want you to notice is the alveoli is surrounded by capillaries, and that makes sense because our capillaries are the blood vessels where exchange takes place, and what we want is we want the air that comes into our alveoli to go into our capillaries, and then the CO2 from our capillaries to go into our alveoli. So about 80 to 90% of the surface of our alveoli are surrounded by capillaries. So why do we branch so much? Again, every time we divide or every time we branch 23 times, we are going to increase the surface area for gas exchange. So at the end where we have our alveoli, that's our 24th division, we have a surface area that's greater than 1 times 10 to the 6. So really, if we were to look at our lung surface area for gas exchange, our alveoli, it could cover the surface of half a tennis court. That's how much surface area we have for gas exchange. And if you recall Fick's law, which we talked about earlier in the term, the greater the surface area for gas exchange, the greater the rate of diffusion. Now let's take a closer look at our alveoli. We have alveoli being made up of 2 types of cells. We have type 1 alveolar cell and type 2. Type 1 alveolar cells are shown here in the purple. So these are the thin purple cells, they're purple in this figure, and this is through which gas exchange will take place. And again, the gas exchange will go from the alveoli into our capillaries, and we can see the capillaries here in pink. The other type of alveolar cell that we have are these yellow guys, and these are called type 2 alveolar cells. And they make an important substance called surfactant. Now, surfactant is going to be really important because it prevents our lungs from collapsing. It decreases surface tension. So we'll talk more about surfactant in a little bit. But first, let me get into gas laws. So what governs movement of air? So we have 3 gas laws I want you to be aware of. The first one is also known as Dalton's law, and it says the total pressure of a mixture of gases is the sum of the pressure of the individual gases. Okay, so what does this mean? It means if you had a grocery bag and I asked you what is the total weight of your grocery bag, and let's say it's filled with apples, bananas, and oranges. Well, then the total weight of your grocery bag would be equal to the weight of your apples, weight of your oranges, and the weight of your bananas all added together. Well, gases exert pressure. So the pressure of a mixture of gases will be the sum of the pressure of the individual gases added together, just like the weight of a grocery bag is the weight of the individual fruits in this case, inside the grocery bag added together. The second law doesn't have a name, so I call it the no-name law, and it says that gases, whether we're talking about a single gas like just oxygen or just CO2, or we're talking about a mixture of gases like the air in the atmosphere, will always move from an area of high pressure to low pressure. So always high to low. And we need that pressure gradient. Without that pressure gradient, we will have no airflow. The third law is Boyle's law. And Boyle's law says that if the volume of a container of gas changes, the pressure of the gas inside that volume will change in an inverse manner. Essentially what that's saying is if we make the volume bigger, the pressure inside will decrease. If we make the volume smaller, the pressure inside will increase. So they're moving in opposite directions. It's an inverse relationship. So let's take a look at how that works. So here we have a container filled with gases. And what we've done is now we've made it smaller. Do you see how we've decreased the volume? And so Boyle's law tells us when we decrease the volume of a mixture of gases, or a container of gases, then the pressure inside will increase. So this container that's half the size will have a greater pressure than this container over here. So now let's say I created a tube, I opened a tube that connected these 2 containers. Which way will my gases move? Remember, the second law that doesn't have a name says that gases move from an area of high pressure to low pressure. Since the beaker on the right has higher pressure, air will move from the right to the left. Now, this is how we breathe. So let's go through that. First of all, we can measure how much air we are moving using a device called a spirometer. You'll actually measure airflow in your lab during one of the BioPack weeks. So a spirometer is going to essentially measure how much air you are moving in and out of your body. When the recording has a positive slope, it indicates inspiration. When it has a negative slope, that indicates expiration. So here is someone who was told to breathe normally. So we have an inspiration, expiration. That is one respiratory cycle. Here is another respiratory cycle. Then what this person did was after a normal expiration, they expired more air. So this was their forced expiration. And then they inspired back up to normal, had one more normal breath, and then after a normal inspiration, they breathed in as much air as they possibly could. Then they breathed everything out that they possibly could, and then they took a couple more normal breaths. So looking at this, we can actually figure out how much air someone is capable of moving and how much air they normally move and what their reserves are. For example, normal breathing, the amount of air that you move every time you breathe quietly or normally or passively, that is known as your tidal volume. And typical tidal volume is 500 milliliters, and that's a number that you should be aware of. So we're going to breathe in 500, out 500, in 500, out 500. Now, the amount of air that you can expire after a normal expiration. So this is a normal expiration, and now I'm going to expire more. So that's going to be the reserve that we can expire. So this is known as our expiratory reserve volume. On the other hand, after a normal inspiration, how much more can I inspire? That's known as our inspiratory reserve volume. That's the reserve or extra we can inspire after a normal inspiration. Another thing that you should be aware of is vital capacity. Vital capacity is how much air are you physically capable of moving. So if I gave you a balloon and I said you have one breath to fill that balloon up with as much air as possible, what would you do? You would take a deep breath in until you can't inspire anymore, which is this point up here, and then you'll breathe all of the air out until you can't expire anymore, and you'll breathe it all into the balloon. So the amount of air that you fill your balloon up with is your vital capacity. That is what you are physically capable of moving. Now, when you breathe all of the air out in your lungs, it doesn't mean there's no more air in your lungs. You actually have air still left over. That's called your residual volume. So this is the amount of air that will never really leave your lungs. And so if we include the residual volume with your vital capacity, that's your total lung capacity. So these are just different lung volumes and capacities that we can measure using a spirometer. Now, men typically have a slightly higher lung volume and capacity than women. Now, another thing that we do in our upper airways, now our upper airways we said is part of the conducting system, the upper airways will also condition the air. So what does that mean? Well, they will warm the air to body temperature so that we don't damage our alveoli. Our alveoli are delicate structures for gas exchange. When we have cold air coming into our alveoli, it can hurt it and damage it. So if you've ever taken a really deep breath in when you were out somewhere really, really cold, you know that it can hurt your lungs. So as the air moves through our upper airways, the upper airways will try to warm the air up to body temperature so that it does not happen. The damage doesn't happen. Another thing that we will do is we'll add water vapor to the air so that we reach 100% humidity so that we don't dry out our alveoli. And then finally, the upper airways, we mentioned this before, will help filter out foreign material to really help protect our alveoli from things like viruses, bacteria, inorganic particles, and other things that we might be breathing in that is not so good for our lungs. Now, we have other things that can help us, or what actually helps us capture those things would be our mucus secreting cells. So in our airways, we have these cells called goblet cells, and these goblet cells secrete mucus. And that mucus is capable of capturing dust particles and other pathogens that may be coming into our airway. And then with the help of cilia, we will then move that mucus and all of the other stuff that it's captured out of our airways. Now, let's take a look at airflow and the things that influence airflow. So airflow is directly proportional to the change in pressure and indirectly proportional to resistance. So that basically means when resistance increases, airflow decreases. When our pressure gradient is higher, airflow increases. Now, when we talk about pressure, what pressure are we talking about? So we have alveolar pressure, which is the air pressure in the lungs, and then we have intrapleural pressure. This is the pressure in the pleural fluid. I'm going to mention the intrapleural pressure, but really the alveolar pressure is going to be the one that we're going to focus on. Now, when we talk about a respiratory cycle, that is one inspiration followed by one expiration. So if air moves from high to low pressure, the reason we inspire is because the alveolar pressure, the pressure in our lungs, is lower than the pressure in the atmosphere. So air goes from the atmosphere into our lungs, high to low. The reason we expire is because our alveolar pressure is now higher than the atmospheric pressure. So air moves from high to low, lungs out. So what causes these pressure changes in our alveoli? Well, movement of the diaphragm and our intercostals, our external intercostals, is one for inspiration. So when we inspire, our diaphragm will actually flatten. Our external intercostals will contract and it'll open up our rib cage and open up our lungs, our chest wall. And so what that does is it increases the volume of our lungs. And remember, Boyle's law says when we increase volume, pressure drops. So that increase in volume drops the pressure in the alveoli. So now alveolar pressure is lower than atmospheric pressure, so air gets drawn in. Now, majority of volume changes occur because of that diaphragm. So that diaphragm is really important in breathing. And again, this is just showing you how our rib cage expands with inspiration. So what is pleural pressure? So pleural pressure is the pressure that's in the pleural sac, so essentially in the pleural fluid. And pleural pressure changes as we breathe, but for our purposes, we're going to say that pleural pressure has a pressure of -3 millimeters of mercury. Now, why is it a negative pressure? Because if you recall, I said your lungs naturally want to collapse. So you see the arrows pointing in. Your rib cage naturally wants to expand. So you see the rib cage pointing out. The pleural fluid acts like glue. So it holds the two membranes together, but it still means the membrane is kind of being pulled apart, if that makes sense. And so what that does is it creates kind of like a vacuum in here, and so we get a negative pressure. If we suffer from a pneumothorax, so a punctured lung, when we rupture that pleural sac and air gets into the pleural sac, then it no longer acts like glue. So the lungs will do what it normally wants to do, which is collapse, and the rib cage will expand. It just can't expand too much because our body is around it. Other things that are important for lung function are the words compliance and elastance. Compliance means you comply really well. You do what you're supposed to do. And our lungs are supposed to be able to stretch. So if you have high compliance, it means your lungs stretch easily. So it's going to be easier to breathe. If you have low lung compliance, that means you have really stiff lungs. Your lungs don't stretch easily. It requires more force. You have to work harder to breathe. Elastance is how elastic are you? And remember, elastic things, not only do they stretch, but after they stretch, they recoil. So do you have that recoil? And for our lungs, we need to be able to comply and stretch easily, but we also need to recoil. So we have to have compliance and elastance so that we can return to our regular or normal volume. There's another law I want you to be aware of, and that is called the law of Laplace. And so the law of Laplace is where we're going to bring in surfactant. So remember, our type 2 alveolar cells secreted surfactant. Now, before we get into surfactant, let's take a look at these bubbles. So here I have a large bubble. Here I have a small bubble. This large bubble has a layer of fluid inside, so does this small one. And what that does is it creates surface tension such that we have a pressure to collapse. So notice these arrows are pointing inward. Now, when we have equal surface tension, a bubble that is smaller will have a greater pressure to collapse. So that's the equation for law of Laplace. So notice the pressure to collapse in this large bubble is 3, and the pressure to collapse in this small bubble is 6. And the only difference between these two is the radius. This one is smaller. So if we look at our alveoli are bubbles. If we had alveoli that were different sizes, which we do, then that means the smaller alveoli would be harder to expand and harder to fill up with air, which really would mean we would be using maybe half our alveoli. All of the bigger ones would get the air and the smaller ones really wouldn't because they're stiffer. So that's where surfactant comes in. What surfactant does is it breaks up surface tension. So we all have surfactant in all of our alveoli, but the larger ones would have less surfactant. The smaller ones would have a lot more. And by breaking up the surface tension, even though this alveoli is smaller, because it has more surfactant and we've broken up the surface tension even more, their pressure to collapse is now equal. And so when air comes in, it will go to both alveoli because both of them will be able to expand with air. So surfactant, again, is more concentrated in smaller alveoli, and it's a mixture of proteins and phospholipids, and it really helps our alveoli it prevents our alveoli from collapsing, and as a result, makes it easier to expand. Now, newborns who have respiratory distress syndrome, what happens is they have inadequate surfactant. So they have really stiff lungs. Their lungs are not very compliant. And so it makes it really difficult to breathe, and the alveoli will actually collapse every time they try to exhale. So what factors affect airway resistance? So the greater the resistance, the less airflow you will have. So the length of your airways will affect it. So the longer the length, the greater the resistance. But in our lungs, that's pretty constant. So it's not a factor we really need to worry about. Another is the viscosity of the air. So the more viscous, the greater the resistance. And it usually is constant, but of course things like humidity, changes in altitude can slightly alter it. But what really impacts airway resistance is the diameter of our airways, or the radius. Radius is just half the diameter. So when we bronchodilate and increase the radius, resistance decreases and airflow increases. When we bronchoconstrict, resistance increases and airflow decreases. So our parasympathetic nervous system causes bronchoconstriction. Our sympathetic causes bronchodilation. So when we have an emergency and we want to run away, that sympathetic will bronchodilate and make it easier for air to flow. Now, other things can, of course, impact airflow. For example, we can have a physical obstruction because of too much mucus secretion. So that's something that we might see in certain conditions like bronchiectasis. Okay, next, ventilation. So ventilation is basically movement of air. So here we have, in this big blue bubble, that's supposed to represent all of your alveoli. And remember, gas exchange is our goal. And gas exchange only happens when we reach the areas of our lungs where gas exchange can occur, and that would be our alveoli. This little square on top represents everywhere else. So our trachea, our bronchial tree, everywhere where air moves within our respiratory tract, but gas exchange does not happen, that's represented by this square, and we're going to call that dead space. And it's dead space because no gas exchange happens in those areas. So we have to start somewhere. So we're going to start with this situation here. So we have some dead space air, right? We have dead space filled with 150 milliliters of air. We have air in our alveoli, and we're going to first expire. We're going to exhale air. And remember, tidal volume, the amount of blood we breathe when we're breathing normally, is 500 milliliters. So when we breathe out 500, this 150 comes out first, and the rest of the 500, the 350, will come out from our alveoli. And that's what we're seeing here. So 150 from the dead space came out, and then 350 from the alveoli came out, and that's 500 milliliters of our tidal volume. Now that we've expired air, we are ready to inspire. Now, when we breathe in air, we're going to breathe in 500. So this new atmospheric air is shown in purple. So here's 500 coming in, but when it comes in, it has to push this dead space 150 down first. So this 150 dead space is pushed down into the alveoli, and then our new air comes in. So what really reaches our alveoli from the new atmospheric air is only 350 milliliters. The other 150 gets stuck in our dead space. So even though we're breathing in 500 milliliters of fresh air, only 350 reaches our alveoli, and so that's what gas exchange is going to occur with. And so when we breathe out, we're actually going to breathe out this fresh air that just came in first, and then some of our stale air, and then it continues on and on and on. So when we look at total pulmonary ventilation, that is, how much air are we moving in and out of our body per minute, we would take our ventilation rate, which is how many times we breathe per minute, and multiply it by our tidal volume, because that's what's coming in and out of our body. But what's more important than what's coming in and out of our body is what's coming in and out of our alveoli. And so when we want to look at total alveolar ventilation, we have to take dead space into consideration. So in this case, we would take our ventilation rate, how many times we breathe per minute, and we would not just multiply it by tidal volume, but we'd multiply it by tidal volume minus the dead space. Because in alveolar ventilation, only 350 is reaching the alveoli. So it's 350 in, 350 out, 350 in, 350 out. Why? Because we have to take the dead space into consideration or out of the equation. So when we're breathing normally, again, our tidal volume is about 500 milliliters. Let's say we do that about 12 times a minute. So if we multiply the two, we have 6,000 milliliters per minute of our total pulmonary ventilation. But if we want our alveolar ventilation, we have to subtract the dead space. So 500 minus 150 is 350. We breathe 12 times a minute, so if we multiply the two, our alveoli is getting 4.2 liters of air per minute. Now let's say we started to breathe shallow. So if we're doing shallow breaths, our tidal volume is going to be much smaller. So let's say it's 300. But because you're moving less air, you'll be able to do it more times a minute. So let's say you did it 20 times. If we multiply the two, what you'll notice is still our total pulmonary ventilation is 6,000. The amount of air that's going in and out of our body is the same. However, let's take our dead space into consideration. So 300 minus 150 is 150. And if we multiply that by 20, we only get 3,000. So even though we're still getting 6,000 liters of air in and out of our body, only 3,000 is going in and out of our alveoli. And remember, it has to reach our alveoli for gas exchange to take place. Now let's breathe deeply. Remember, slow, deep breaths. That's what you're told to do when you start to panic. So when you do slow, deep breaths, let's say we're moving 750 liters of air every time we breathe. Now, you're not going to do it as quickly, so let's say you only do 8 breaths a minute. If we multiply the two, we still have the same 6,000 liters of air. Now, we have to subtract the dead space, so 750 minus 150 is 600. 600 times 8 is now 4,800. So by breathing slowly and deeply, we actually get more air into our alveoli. And that's all because dead space doesn't change. It stays 150. And that's pretty constant from person to person. Now, based on height and things like that, it might be slightly different. But for our purposes, it's going to stay 150. Now, we can also change our pattern of ventilation. I'm just going to focus on these two, hyper and hypoventilation. So in hyperventilation, we're going to increase the amount of air that we are moving in and out of our body. And in hypo, we would decrease that. And these are the description and some examples. So to summarize, our total pulmonary ventilation in a normal individual, so average number is 6 liters, so 6,000 milliliters per minute. And again, total alveolar was 4,200 or 4.2 liters. And I got that from right here, okay? And if we were to push our lungs to the max, that's our maximum voluntary ventilation. We could go from just moving 6 liters of air per minute when we're breathing normally to breathing 125 to 170 liters per minute. And that's why we're able to exercise, because we can increase our ventilation. And again, our respiratory rate is between about 12 to 20 breaths. Okay, so now, when we're breathing normally, what is the level of gas in our alveoli? So when we're breathing normally, which is moving about 4,000, right, 4,200 to be exact, milliliters of air per minute, if we were to measure the partial pressure of oxygen in the alveoli, it's going to be 100 millimeters of mercury. So it's going to be very oxygen rich. CO2 levels would be about 40 millimeters of mercury. Now, that's normal. So 100 for oxygen, 40 for CO2. From normal breathing, if we were to hyperventilate, when we're hyperventilating, we are moving more air. Then we are going to bring in more oxygen and breathe out more CO2. Remember, when we ventilate, we're breathing in more oxygen, breathing out more CO2. So the more you hyperventilate, partial pressure of oxygen in the alveoli will rise and the partial pressure of CO2 will fall. On the other hand, if you hypoventilate, you move less air, you're going to bring in less oxygen and you're going to breathe out less CO2. So then oxygen levels will drop and CO2 levels will rise. Now, the last thing I want to mention is that we have to match ventilation with perfusion. So ventilation is the movement of air. Perfusion is the movement of blood. Why do we have to match it? Because the air that we bring into the alveoli, we have to then get into our blood. So in this example, we have 2 alveoli that are getting ventilated, and they're both getting a blood supply. So we have deoxygenated blood coming and oxygenated blood leaving. If we had a problem with this alveoli on the right, let's say we have a bronchoconstriction, an obstruction, too much mucus. For whatever reason, this alveoli is getting hypoventilated. Remember, when we hypoventilate, oxygen drops, CO2 rises. So notice, partial pressure of CO2 went up, partial pressure of oxygen went down. If we send blood equally to both alveoli, the blood over here will pick up oxygen and give away CO2, but the blood over here, it doesn't have oxygen to pick up. And because CO2 is high in this alveoli, what might even happen is my blood might pick up CO2. Because remember, gases just move from high to low pressure. So what's going to happen is we're only going to oxygenate half our blood supply. So what we're going to do to respond to this is match ventilation and perfusion. So since this alveoli is not getting ventilated, we are not going to perfuse it. So let's vasoconstrict the blood going to the alveoli over here. And by doing that, we will redirect blood to this blood vessel, and so more of our blood has the potential to pick up oxygen and remove CO2. And so this is one way our body responds when some of our lungs start to get hypoventilated. Okay, thank you.